Welcome BioSquad. This one right here is a different video from the rest of the series. This is a 9700 Specimen Paper 1 for examination from 2022. If you are new to this channel, you know what to do. Like, comment, subscribe, and finally watch and learn. Without wasting further more time, let's get into it. Question 1. Which definition of the magnification of a drawing of a leaf is correct? A. The actual size of the leaf multiplied by the magnification of the microscope. B. The measured difference in size between the leaf and the drawing of the leaf. C. The increase in size of the leaf when observed using a microscope. D. The size of the drawing of the leaf divided by the actual size of the leaf. When they ask for the definition of magnification, you should always keep in mind the formula, which is magnification is equal to image size upon actual size. And if you go to the answers A, B, C and D, the best possible choice being option D. Size of the drawing of the leaf, that is the image size, divided by the actual size. Hence, D is the correct choice. Question 2. The electron micrograph shows part of two cells. Which label features identify these cells as eukaryotic? If you go to this electron micrograph, part X doesn't help in identifying cells as eukaryotic. Why is that? Because part X represents a cell wall, and cell walls can be present in eukaryotic cells as well as in prokaryotic cells. Because cell walls are present in prokaryotic cells too. It is just the substance that differs which makes the cell wall. Therefore, if you go to the answers A, B, C and D, since answers A, B and D have X in it, they are incorrect, leaving us with the best possible answer being C, W only. And W is nothing but chloroplast. Question 3. Plant cells are stained and viewed using a student microscope. The light source is natural light. What could be clearly visible at 400 magnification? This question seems to be very common in most of the papers, and if they mention light microscope and if you have practiced viewing of slides, the only possible structure clearly visible is the nucleoli. Therefore, the answer is C. The structure that is clearly visible at times 400 magnification would be the nucleoli. Question 4. Which lengths are equivalent to 1 micrometer? 1 micrometer is equal to 1000 nanometers as well as 1 micrometer is equal to 0.001 millimeter, as 1 millimeter is equal to 1000 micrometers. So you simply have to apply the math over here and convert from one unit to the other, leaving us with the answer being D, 3 and 6. 1 micrometer is equal to 1000 nanometers, which is obviously correct. Therefore, the answer should have 6 in it. And if you go to the answers A, B, C and D, the only possible answer that has 6 in it is D. And yes, 3 is also correct because 1 millimeter is equal to 1000 micrometers. Hence, 1 micrometer will be equal to 0.001 millimeter. Just apply the math and this question is nothing but easy. Hence, the answer is D. Question 5. Some secretory cells synthesize and release glycoproteins. What is the correct sequence of some of the events that occur in a secretory cell? 1. Exocytosis. 2. Products accumulate in secretory vesicles. 3. mRNA binds to ribosomes. 4. Synthesis of glycoproteins. First, the protein should be made and then it will be modified. Therefore, protein synthesis occurs in the ribosomes, which should start with 3. mRNA binds to ribosomes, leaving us with the answer being either A or B. And following protein synthesis would be protein modification. And where does protein modification occur? It occurs in the Golgi body and it forms glycoproteins. Therefore, the next step would be 4. Synthesis of glycoproteins. Then after it is modified, then comes the releasing done at the very end. The process of releasing is known as exocytosis, and exocytosis would obviously be the last stage. And in order for exocytosis to occur, the product should accumulate in secretory vesicles because it is the vesicles that go and fuse with the cell membrane, allowing the contents to leave the cell. Therefore, the process exocytosis should appear last, leaving us with the answer being B. First is mRNA binds to ribosomes, followed by synthesis of glycoproteins, followed by products accumulate in secretory vesicles, and lastly exocytosis. A scientist used a blender to break open a sample of animal cells and release the organelles. The extracted mixture was spun in a centrifuge at a low speed. The heaviest type of organelles sank to the bottom of the centrifuge tube to form a solid pellet, pellet 1. The liquid above pellet 1 was poured into a clean centrifuge tube and spun at a higher speed. The next heaviest organelles sank to the bottom to form a solid pellet, pellet 2. The scientists repeated this procedure twice more to obtain pellet 3 and pellet 4, each containing a single type of organelle. What is a function of the type of organelle extracted in pellet 3? So obviously the pellets obtained were in the order from heaviest to lightest, and the heaviest would be the nucleoli and the lightest would be the ribosomes. If you go to the order, it would be first the nucleoli, followed by the mitochondria, then the lysosomes, then the endoplasmic reticulum, and lastly ribosomes. Since they are asking for the organelle extracted in pellet 3, that should be, if you go from the left of this order, that would be lysosomes. Pellet 3 is lysosomes, and what do lysosomes do? They carry out the digestion of all cell organelles. Therefore, the function of pellet 3, that is a lysosome, is A. Digestion of all cell organelles. Question 7. What is the general formula for amylose? Amylose is a polymer of alpha glucose, hence it will have 6 carbon atoms, and it is formed by a process known as condensation, where a water molecule is removed or it is formed. And if you go to the answers A, B, C and D, answers A and B are incorrect because they have 5 carbon atoms. However, the answer should have 6 carbon atoms. And if you go to options C and D, why is C incorrect? Because it mentions C6H12O6. However, when a water molecule is removed, that is H2O, it will be C6H10O5, not H12O6. Therefore, the answer is D, C6H10O5. Question 8. Which statement describes how the molecular structure of starch is suited to its function? A. Amylose has a branch structure and amylopectin is coiled to give a compact molecule for transport. B. In the breakdown of amylose and amylopectin, many condensation reactions release stored energy. C. In the formation of amylose and amylopectin, many hydrolysis reactions allow the release of stored energy. D. Amylose and amylopectin form a complex that is insoluble and so does not affect the water potential of the cell. Always remember this carefully. Formation is related to condensation reactions and breakdown is related to hydrolysis reactions. And amylopectin is a branched molecule, whereas amylose is a linear molecule and hence it is not branched at all. And if you go to the answers A, B, C and D, why is A incorrect? Because it mentions amylose being branched. However, amylose is not branched, therefore A is incorrect. B. Many condensation reactions release stored energy. In the breakdown, it is hydrolysis, not condensation. As it mentions in the breakdown of amylose and amylopectin, it should be hydrolysis, therefore B is incorrect. C. In the formation, many hydrolysis reactions, that is also incorrect because formation is related to condensation reactions. Therefore, leaving us with the only answer being D. Amylose and amylopectin form a complex that is insoluble, yes, and so does not affect the water potential of the cell. That is correct. Question 9. Cows and whales are mammals that produce milk to feed their babies. Newborn whales grow faster than newborn cows. Milk contains fatty acids with a range of chain lengths. The table shows a percentage of fatty acids of different chain lengths in cow milk and whale milk. Which statement about the ratio of short fatty acids, that is 4 to 16 carbon atoms, to long fatty acids, that is 18 or more carbon atoms in the milk of cows and whales is correct? You simply have to go to the percentages given in this table and then check out the ratios between the two, that is in cows and whales. In cows, the short chains percentage is 58.3, long chains percentage is 41.7. In whales, the short chains percentage is 41.7 and the long chains percentage is 58.3. Therefore, the ratio in whale milk is lower, hence the answer being either A or D, because A mentions the ratio in cow milk being higher, which means that the ratio in whale milk is lower, and D mentions the ratio in whale milk being lower. And a key thing you should know is that the longer the chains of fatty acids, the more carbon to hydrogen bonds present. So more energy is available in newborn whales than newborn cows. Why is that? Because in newborn whales, the percentage of long chains is greater than the percentage in cows. As you can see, 58.3% is greater than 41.7%. Therefore, the answer being D. The ratio in whale milk is lower because newborn whales need more energy than newborn cows. Over here, you simply have to go through the percentages, check which ratio is lower, then finally come to a conclusion based on which one has longer chains, and the one having longer chains will have more energy available because longer the chains of fatty acids, the more carbon to hydrogen bonds available. Therefore, the answer is D. Question 10. The structure of phospholipids includes 1. Polar phosphate heads, 2. Hydrophobic fatty acid chains, 3. Saturated fatty acid chains. What are essential for the formation of a phospholipid bilayer in a cell surface membrane? A phospholipid bilayer consists of polar phosphate heads and hydrophobic fatty acid chains. The fatty acid chains can be either saturated or unsaturated. It doesn't have to be always saturated fatty acid chains. Therefore, answers having 3 in it are incorrect, leaving us with the answer being A, 1 and 2 only. Again, why is 3 incorrect? Because 3 mentions saturated fatty acid chains, and that is not essential because fatty acid chains can either be saturated or unsaturated. However, the other two are important in the formation of a phospholipid bilayer. Question 11. Diagram shows a molecular structure of a peptide. Which molecules will result from the complete hydrolysis of the peptide? Hydrolysis occurs across the C double bond O single bond NH group, and there will be no other changes to the other groups, and hence the other groups remain intact as no changes in bonds. Again, hydrolysis occurs across the CONH group, and I have marked over here where water would be added in order to break that bond, and as you can see over here, 2 water molecules are needed to break those 2 C double bond O single bond NH bonds. And once those 2 bonds are broken, and if you carefully go to the resulting molecule along with the answers, you would get the answer being C. Again, the other groups remain intact, no changes occur to them. Therefore, C represents the best possible answer, as no changes occur to the other groups and only across the CONH group. Question 12. The diagram shows a hemoglobin molecule. Which row identifies the different parts of the molecule? Now, hemoglobin is a globular protein, hence it is soluble in water as it has hydrophilic groups pointed towards the outside regions and hydrophobic groups towards the center. And if you go to the answers A, B, C and D, the answers include hydrophilic as well as hydrophobic amino acid chains. And if you go to this given diagram, where should hydrophilic amino acids lie? It should obviously lie towards the outside region because it is soluble in water. And what do you mean by hydrophilic? Hydrophilic means water loving. Therefore, which chain represents the outer regions? It is either number 2 or number 3. And which one represents hydrophobic amino acids? It should be definitely number 4 because only number 4 points somewhat towards the center of the hemoglobin molecule. Therefore, 4 is definitely hydrophobic amino acids. And if you go to the answers, the answer being either A or B. And if you look at 1 seems to be different in appearance compared to the others because 1 seems like a sort of a circular shape, whereas the others are represented as coils. Therefore, 1 should definitely be oxygen binding site and not alpha helix. Therefore, the answer being B. 1 oxygen binding site, 2 hydrophilic amino acids, 3 alpha helix, 4 hydrophobic amino acids. Question 13. A student investigated the effect of enzyme concentration on the rate of hydrolysis of protein in milk. When the enzyme and milk were mixed, the protein was hydrolyzed and the mixture changed from cloudy to clear. The student investigated 5 different enzyme concentrations and recorded the time taken for the mixture to become clear. What is an appropriate control for this investigation? The reaction occurs to show the hydrolysis of milk and hence the color change. Therefore, a control would be to observe in water solution and whether any color change occurs. Therefore, the best possible answer being A. Carrying out an experiment where the enzyme solution is replaced with water, because enzymes are needed to carry out the digestion of the milk and hence the color change. Without the enzyme solution, there would be no reaction as such occurring. Therefore, the appropriate control would be replacing the enzyme solution with water and obviously we can conclude the fact that there won't be any color change occurring when there is no enzyme present, and that is an appropriate control. Therefore, A is the correct answer. Question 14. What determines the specificity of an enzyme? Over here, specificity is determined by an enzyme's tertiary structure always, hence the bonds between the R groups of the protein molecule, because the bonds holding the R groups together determines the tertiary structure. Therefore, the best possible answer being C, 1 only. The covalent and other bonding between R groups of the polypeptide. As only statement 1 involves R groups in the answer, the other statements are not necessary to determine the specificity of an enzyme. Hence the answer is C, 1 only. Question 15. The graph shows the effect of temperature on the rate at which the enzyme in a biological washing powder digests and removes fruit juice stains. Which statement explains the shape of the graph at temperatures higher than T? At temperatures higher than T, enzyme's active site changes shape due to enzyme denaturation. This we can obviously infer from this given graph, because at temperatures higher than T, the rate of reaction steeply decreases till it reaches 0, and that represents enzyme denaturation. Enzyme denaturation, if it occurs, leads to active site being altered, and if the active site is altered, the tertiary structure is altered too. Therefore, the shape of the active site and substrate are no longer complementary. And if you go through the answers, 1. Bonds are broken between the R groups of the amino acids in the polypeptide chains of the enzyme. Yes, 1 is a possible statement, because bonds holding the R groups represents tertiary structure, and the tertiary structure is affected if the active site changes shape. Therefore, 1 is correct. 2. There are more collisions between the enzyme and its substrate. That doesn't necessarily explain the shape of the graph at temperatures higher than T. Let's look at 3. The tertiary structure of the enzyme is altered. Yes, that is correct, because bonds between the R groups being broken leads to the tertiary structure being altered. Therefore, 3 is correct. 4. The shapes of the active site and substrate are no longer complementary. That is also correct. Due to enzyme denaturation, active site of the enzyme changes shape. If the active site changes shape, the substrate is no longer complementary in nature to fit with the active site. Therefore, the best possible answer being C, 1, 3 and 4. Question 16. Which statement describes carrier proteins in cell surface membranes? A. Carrier proteins are found on the outer surface of the membranes, allowing cell recognition. B. Carrier proteins are involved in moving substances through the membranes by the active transport or passive transport. C. Carrier proteins allow the binding of ligand molecules, which causes changes within cells. D. Carrier proteins are involved in moving substances through the membranes by passive transport through water-filled pores. Carrier proteins are involved in transport across membrane. Hence, the answer being B. Carrier proteins are involved in moving substances through the membranes by either active transport or passive transport. Where are carrier proteins used in passive transport? It is used in facilitated diffusion, because facilitated diffusion is a passive process that moves substances through the membrane using carrier proteins. And active transport does obviously use carrier proteins in their process. Therefore, answer B is the correct choice. Carrier proteins are involved in transport across the membrane in moving substances. Question 17. What happens to a typical bacterium when it is placed in surroundings that have a higher water potential than the water potential inside the cell? Higher water potential outside the cell means that the water will enter the cell down a water potential gradient, because higher the water potential in the surroundings, more water molecules present that will enter down a water potential gradient from higher water potential to lower water potential. And if the surroundings have higher water potential, that means the water potential inside the cell will be lower. Therefore, there will be a water potential gradient set up, and water will enter the cell from outside to inside. And once water enters the cell, what happens? The bacterium does not burst, because a typical bacterium has a cell wall, and cell walls doesn't allow bursting. Therefore, A is incorrect. B. The bacterium dies because water leaves the cell. That is incorrect, because water doesn't leave the cell. Water rather enters the cell. C. There is no change because the cell wall is impermeable to water. That is incorrect, because cell wall is freely permeable in nature. It will allow any substance to pass through it. Hence, leaving us with the best possible answer being D. There is a net movement of water into the bacterium, which is correct. Question 18. By which process do hydrogen carbonate ions leave lead-bread cells? Always remember that hydrogen carbonate ions leave the cell by facilitated diffusion. Hence the answer being C, facilitated diffusion. Question 19. In an experiment, fluorescent dyes were attached to proteins on the outer surface of cell surface membranes. Fluorescent dyes of one color were attached to proteins of a living human cell, and fluorescent dyes of a different color were attached to proteins of a living mouse cell. The human cell and the mouse cell were then fused to form a hybrid cell. At first, the proteins attached to different fluorescent dyes remained separate, but after 40 minutes, the proteins were distributed randomly across the hybrid cell surface membrane. What does this experiment show? This experiment supports the fluid mosaic model and mainly the fluid nature, because it clearly shows that the proteins are able to move and diffuse within their monolayers. Hence the answer being C. Proteins move freely in the phospholipids of a bilayer, because fluid refers to the proteins and phospholipid molecules being able to move, and in order for proteins to be distributed randomly after 40 minutes, it does show that the proteins move freely. Therefore, C is the correct choice. The other choices are incorrect because it does not support the observations obtained from the experiment. Question 20. The diagram shows a mitotic cell cycle during which phase is DNA replicated. DNA replication always occurs in the S phase of interphase. And if you go to this given diagram, starting from cytokinesis, then it will be followed by G1 phase. After G1 phase would be the interphase, and then the G2 phase, and then finally mitosis. And where is DNA replicated again? DNA is replicated in the S phase of interphase. Since there is no S phase but interphase is given, the answer is B, interphase. Question 21. Bacterial cells divide by a process called binary fission. Which macromolecules must be synthesized for binary fission? 1. Cell membrane proteins and RNA. 2. DNA and peptidoglycan. 3. Enzymes and cellulose. Enzymes and cellulose is obviously incorrect because it consists of the word cellulose. And cellulose, if you recall correctly, is only present in plant cells, not in bacterial cells. Therefore, answers having 3 in it are incorrect, leaving us with the answer being B, 1 and 2 only. Cell membrane proteins and RNA as well as DNA and peptidoglycan are necessary for binary fission. Question 22. The electron micrograph shows a group of human chromosomes. Which label is correct for each of the structures labeled W, X and Y? W represents nothing but the centromere, as it is what holds the two chromatids together. X represents a telomere, as they are always found towards the ends, and Y is nothing but the chromatid. Therefore, the answer being C, W being the centromere, X being the telomere, Y being the chromatid. Question 23. Which statement about the behavior of chromosomes during mitosis is correct? A. They attach to the spindle fibers to contain them within the nucleus. B. They condense to prevent further translation of genes. C. They reach the poles of the cell and begin to uncoil. D. They replicate to produce sufficient DNA to form two nuclei. Replication doesn't occur in mitosis, therefore D is incorrect. And A and B doesn't talk about the behavior of chromosomes during mitosis and are somewhat incorrect statements. C is a correct statement. They reach the poles of the cell and then they begin to uncoil. And that process is known as anaphase, where anaphase is where the chromosomes reach the poles of the cell, and following anaphase is telophase, where it begins to uncoil. Therefore, C is the correct choice. Question 24. Gene mutations can occur by substitution, deletion or insertion. What is the smallest part of a DNA molecule that can be changed by a gene mutation? Definition of a mutation, if you recall, is a change in the sequence of nucleotides. And a DNA molecule is made up of nucleotides. Hence, what is changed by a gene mutation is a nucleotide, because mutation is again a change in the sequence of nucleotides. And since the question refers to the smallest part that can be changed by gene mutation, that is nothing but answer D, nucleotide. Question 25. Which statements about mRNA are correct? 1. mRNA can form base pairs. 2. mRNA can form hydrogen bonds. 3. mRNA is single-stranded. mRNA is definitely a single-stranded molecule, and all the given statements do apply. They can form base pairs and they can form hydrogen bonds, because hydrogen bonds are involved in forming base pairs. Therefore, the answer being A, 1, 2 and 3. Question 26. The table shows the role of 4 different proteins involved in DNA replication. The proteins given are helicase, which unwinds the parental DNA double helix, topoisomerase, which breaks and rejoins the DNA strands, single-stranded binding protein binds to separated DNA strands to stabilize them, DNA polymerase synthesizes strand of DNA. Which row shows the function of these proteins? Protein helicase causes the unwinding. Therefore, unwinding leads to producing 2 template strands. Hence, the answer being D, makes strands available as templates, as that is the best possible explanation. And if you look at DNA polymerase, the role being to synthesize strands of DNA. What is the function of DNA polymerase? Well, DNA polymerase adds nucleotides to the end of a growing polynucleotide strand. Therefore, yes, D is a correct choice. Helicase makes strands available as templates. Topoisomerase enables tension caused by unwinding to be released. Single strand binding protein prevents original strands reforming complementary base pairs. DNA polymerase adds DNA nucleotides to the 3' end of a growing polynucleotide strand. Again, this question would be easy by just ruling out the given options, because obviously DNA polymerase adds nucleotides to a growing polynucleotide strand, hence being either B or D. And the function of helicase is to unwind the parental DNA double helix, and once unwinding is caused, that will lead to a formation of 2 template strands, and the answer that involves template strands is answer D. Therefore, the best possible choice being D. Question 27. Which type of sugar and which type of bond are found in a DNA molecule? DNA is made up of a ribose sugar, which is a monosaccharide, and the bonds present in monosaccharides are not peptide at all, as peptide bonds are found in proteins, not in sugars. Therefore, C is obviously incorrect because it mentions peptide bonds, which is not present in sugars. And the type of sugar is monosaccharide, hence the best possible answer being D. Type of sugar monosaccharide, type of bond hydrogen. A and B are incorrect because the type of sugar is not monosaccharide, and C is incorrect because the type of bond given is peptide. Therefore, D is the best possible answer. Question 28. Diagrams represent transverse sections of 3 plant organs. Which row is correct for phloem? I have labeled towards the sides which one represents which. 2, 3 and 6 represent the phloem. 1, 4 and 5 represent the xylem. And since the question refers to which row is correct for phloem, that should be 2, 3 and 6 only. And if you go through the answers, the only answer that has 2, 3 and 6 in it is C. For root it is 2, for stem it is 3 and for leaf it is 6. Question 29. Sucrose moves from a phloem sieve tube element into a root cell. Which changes to the water potential and the volume of liquid in the phloem sieve tube element are correct? This question right here mentions the sucrose leaving a phloem sieve tube element, hence the water potential inside a phloem sieve tube element should rise, or it should become higher. Because if sucrose is leaving a phloem sieve tube element, then the water potential inside the phloem sieve tube element will become higher. As you recall the process of loading and unloading of sucrose, if you load sucrose into a phloem sieve tube element, the water potential decreases. If unloading occurs, that is sucrose leaving a phloem sieve tube element, the water potential inside the phloem sieve tube element becomes higher. Hence, the answer being either A or B, water potential becomes higher. And a key thing you must note is that if water potential becomes higher, the volume decreases, and vice versa. Therefore, the best possible answer being A. Water potential becomes higher, volume of liquid decreases, as they have an opposite correlation. Question 30. Which statements about water movement in plants are correct? 1. Water can pass through cell walls containing layers of cellulose fibers. 2. Water can pass through cell walls containing rings or spirals of lignin. 3. Water cannot pass through a band of suberin in cell walls. 3 is obviously correct because suberin deposits prevents the apoplast pathway which involves the water movement. Therefore, water cannot pass through at all if there is a band of suberin in the cell walls. Therefore, answer should have 3 in it, and 1 and 2 are also correct because cell walls are freely permeable, and since 1 and 2 both talks about the movement through cell walls, yes, water movement does occur through the cell walls. Therefore 1, 2 and 3 are incorrect, leaving us with the answer being A, 1, 2 and 3. Question 31. The photomicrograph is a transverse section of a leaf. Which labeled features are characteristic adaptations of many xerophytes? Xerophytes, they obviously have sunken stomata, trichomes or hairs on its surface, as well as they have a thick upper epidermis and a waxy layer. And the labeled features that are characteristic adaptations would be obviously 1, 2 and 5. Why is that? Because 1 represents sunken stomata, 2 represents the trichomes or the hairs on its surface, 5 represents the waxy layer. 4 and 3 doesn't really represent any characteristic adaptation of many xerophytes. Therefore the answer being only C, 1, 2 and 5. Question 32. A maize crop is growing successfully in a field in which the water potential of the soil is minus 40 kilopascals. What is the most likely water potential of the cell sap in the root hair cell? Water potential in the root should be lower than soil so that water enters the cell. In other words, water potential in the soil should be higher than the water potential in the root hair cell. And since the water potential of the soil is already given as minus 40 kilopascals, more negative the number means lower the water potential. And since the cell sap in the root hair cell should have a lower water potential than the water potential of the soil, it should be a more negative number than minus 40 kilopascals, because once again, more negative the number, lower the water potential. Therefore the best possible answer being minus 60 kilopascals, because the other answers are either equal or less negative than minus 40 kilopascals, and that is not what we need. We need a more negative number in order for the water potential of the cell sap in the root hair cell to be lower than the water potential of the soil. And minus 60 is obviously more negative than minus 40, hence is the most likely water potential of the cell sap in the root hair cell. Question 33. The contraction of the heart is coordinated through electrical impulses passing through the cardiac muscle. What is the correct order of part of the sequence of these impulses? It obviously starts with the sinoatrial node sending waves of excitation, therefore being either B or C. Following sinoatrial node, it should be the right and left atria, which is present in both B and C. Then it is obviously the atrioventricular node, not the Purkinje tissue, because the atrioventricular node causes a delay and then sends the impulses through the Purkinje tissue, not the Purkinje tissue sending impulses to the atrioventricular node. Therefore C is incorrect. The best possible answer being B, sinoatrial node followed by right and left atria, then the atrioventricular node which causes the delay and finally sends the waves of excitation down through the Purkinje tissue, and Purkinje tissue should be at the last, hence the answer is B. Question 34. Which features allow the aorta to withstand high blood pressures? Over here you simply have to keep in your mind that the features that allow the aorta to withstand high blood pressures are collagen fibers as well as elastin fibers, not anything else out of the given options. And over here you simply have to memorize it. Hence the answer is A, collagen fibers and elastin fibers, where elastin is necessary for the stretch and recoil function of the aorta. Question 35. Which row is correct for the pulmonary artery? All arteries carry oxygenated blood apart from the pulmonary artery, as pulmonary artery is an exception. Therefore, the blood carried in the pulmonary artery should be deoxygenated, because again all arteries except pulmonary artery carry oxygenated blood. Hence the blood in pulmonary artery would be deoxygenated, and all arteries do have a narrow lumen or a small lumen size. Therefore the answer being D, blood carried deoxygenated, thickness of wall thick, lumen size is small. Question 36. The graph shows oxygen dissociation curves of adult hemoglobin in different carbon dioxide concentrations 1 and 2. Which conditions could change the shape of curve 1 to shape of curve 2? The curve shifts to the right from 1 to 2. When it shifts to the right, it indicates that there is a higher concentration of carbon dioxide. Hence the answer being either A or B, because A or B mentions increased carbon dioxide concentration, which is correct as it causes the shape of the curve to shift from 1 to 2. Increase in carbon dioxide leads to a lower pH due to formation of carbonic acid, and dissociation to H plus ions leads to lower pH as well, and increase in H plus means lower pH. Therefore, the answer being A. Increased carbon dioxide concentration causing a decrease in pH, because after carbon dioxide concentrations increases, with the help of an enzyme known as carbonic anhydrase, it will form carbonic acid, and all acids do cause a decrease in pH. Therefore, A is the correct choice. Question 37. What maintains the necessary concentration gradients for carbon dioxide and oxygen in the lungs? Concentration gradients is maintained by ventilation, as inhaling leads to providing oxygen and exhaling releases the carbon dioxide buildup inside the body. This thereby leads to reducing the carbon dioxide concentration in the alveoli whilst increasing the oxygen concentration. Therefore, out of all the given options, the best possible answer being A, good ventilation of the lungs. When you breathe in, oxygen is supplied to your lungs, and when you exhale, carbon dioxide buildup inside your lungs is released, and that maintains a concentration gradient. Hence, A is the correct choice. Question 38. Which cells and tissues are present in the trachea? Trachea has cartilage, ciliated epithelium as well as goblet cells. And as a side note, cartilage is present in the form of C-shaped rings in the trachea, whereas in the bronchus it is present in the form of irregular blocks or irregular plates. However, since the question refers to which cells and tissues are present in the trachea, it should be B. Cartilage, ciliated epithelium as well as goblet cells are found in the trachea. Question 39. The global mortality figures for some diseases in 2013 are shown in the table. How many millions of people died in 2013 from the bacterial diseases listed in the table? HIV/AIDS. HIV/AIDS is caused by a virus, not a bacterium. Therefore, it is not a bacterial disease. TB is caused by a bacterium known as Mycobacterium tuberculosis or Mycobacterium bovis, and malaria is caused by Protoctis, not a bacterium, therefore is not included as a bacterial disease, whereas cholera is definitely caused by a bacterium known as Vibrio cholerae. And since the question refers to how many people died from bacterial diseases, the answer should be 1.40, that is of TB, plus 0.02, that is of cholera, which gives you the answer as 1.42 million. Hence, the answer is B. Question 40. Why is it difficult for B lymphocytes and T lymphocytes to respond to the antigens on pathogens that are intracellular parasites? If they are within cells, antigens are hidden or concealed. Therefore, lymphocytes are unable to attack body's own cells that hides the pathogens. Hence, the lymphocytes do not encounter the antigens. And if you go to the answers A, B, C and D, the only answer that talks about this difficulty is answer C. The lymphocytes do not encounter the antigens. Answers A, B and D are incorrect and do not explain the difficulty for B lymphocytes and T lymphocytes to respond to the antigens that are intracellular parasites. Intracellular meaning acting within the cells or hiding inside cells. And if the antigens are hidden or concealed, lymphocytes cannot attack these antigens because they are hidden inside body cells, and lymphocytes do not attack our own body cells. Therefore, it does not encounter the antigens at all. Hence, the answer is C. The lymphocytes do not encounter the antigens. And that is the end of this paper. Hope you did find this video useful. If you did, leave a like and do not forget to subscribe if you haven't already. Please do share it with your friends, and let's keep this bioscore growing. Thank you and stay tuned for more.